We are having some special guests from PerMedCoE Center of Excellence, and they are part of the Barcelona Supercomputing Center, and they will be presenting their talk on life science. You can start, Alfonso.
Thanks. I was waiting for not knowing for what. So my name is Alfonso Valencia. I'm the director of the Life Science Department of the Barcelona Supercomputing Center, and also the PI of this new Center for Excellence on Personalized Medicine. I have with me Arnaud Montague, that is responsible of a good part of the organizing of this Center of Excellence. What we are going to do is I'm going to make a relatively short introduction to why we started this new Center of Excellence and what is the scientific and technical point of what we are doing. And then Arnaud will be going into the details of some of the things that we are doing and show some of the results and some of the processes. Let me share the screen for starting. Yeah, so as I say, simulations in molecular biology and biomedicine, that's the focus of the PerMedCoE. So the dream for the future of medicine and how we fit this idea in a general framework of the future is this idea of digital twins. Digital twins, as you probably know, are a frequent tool or system in many areas of engineering. In this case, thinking on digital twins for medicine will be following this sort of ideal schema. Anna has some type of condition or an accident or disease, goes to a medical center where a number of data has already been collected, the genomic information, the devices, the gadgets, clinical tests, and all this information is sent back to the virtual Anna that lives in the virtual world, the digital twin of Anna, where a model of Anna has been already used for preparing conditions. What will happen if Anna has this and that, and is updated with the new information? A number of experiments are done in a computer, and a number of recommendations are sent back to the real world for Anna and the clinicians to take decisions about this possible condition. That's ideal. In other areas of engineering, this can be done. In biology, it's more difficult because we don't know enough about the system Anna to have a model of the full body of Anna with the different physiological conditions. Humans or living systems are too complex at this point to have these type of models. But we are making progress as the general area of research, and you have seen these simulations. These are molecular dynamic simulations, a receptor and a protein and a ligand, a potential drug, trying to find a binding site. So this atomic steel level, we already have tools that are getting more and more powerful to do simulations. At the other level of organs, we start having tools that are able to reproduce physiological behaviors, again, in a complex systems, organs in this type. The purpose of the PerMedCoE was to cover this gap between these molecular simulations and these organ simulations. What is in between? In between is what is going on inside the cell, the molecular systems that are working inside each one of the cells of our body, meaning different proteins and compounds, chemical compounds and RNAs interacting together to make the physiology of the cell work, and then all these cells interacting one with the other, forming these organs, a tumor, a an embryo during the development, and then being able to simulate this multicellular system. So essentially, PerMedCoE is something that fits in between the atomistic level, the organ level, bringing these intracellular and intercellular levels. Why to do this now and not 5 years ago? And I think there are two key experimental issues, two key techniques that makes this possible now following these new developments. One of them is single cell genomics. Until now, when we were analyzing a tumor, for example, what the biologists were doing was to take a piece of the tumor with a few millions of cells and analyze this as a single entity, as the average. Now we have technology to do single cell genomics, meaning being able to see what is the level of activity at the genomic level of individual cells. In this case, this tumor is one of the dots. Different colors will be a different cell with their own behavior, with their own state of physiology, in this case, RNA expression, for example. So this allows us to have cells in a system, in a model, with different experimental information that are reflecting a real physiological state, something that was not possible, let's say, 5 years ago. The other contribution from the experimental side are organoids. Organoids are small organs, as the name indicates, in which the basic aspect of the physiology are reproduced, meaning this can be grown and different cells will be taking different roles, being the epithelium, being the internal part, being the building at the 3D level and also at the specific functional level of the different organs. So now we can imagine simulating something that is for which we have an experimental control. We can have an organoid representing a given human organ. The brain will not be as complex as the brain, but will be reproducing some basic functions of the brain. And we can have this with different mutations in different conditions or different drugs in a way that this will be the experimental equivalent to things that we can simulate. So this is actually, so this is what we are trying to do, to provide a mechanistic description of the processes that goes from the single cell level to the at least organoid level, covering this area of knowledge. Is this just dreaming or is this working? The example is from COVID-19. There is a big community putting together all the information about what we know about COVID-19 at the molecular level. This means understanding how much do we know about the interaction of each one of the viral proteins with the human proteins. This is the collection of viral proteins, it's relatively small, and these are the experimentally assessed interactions between the viral proteins and the human proteins. And now we can put this in maps. This will be a map of a normal human behavior, and this is how the viral proteins are interacting with different parts of the human system. So this is a map of the interactions at the molecular level between human proteins and viral proteins. So now we can, in principle, we can use this information to inform different cells of the human body that are infected by the virus, and then make simulations of what will happen in different situations with different drugs or with different cell types, because at the same time, there are many, in this representation, the blue cells will be epithelial cells, the green cells are cells from the immune system, and they have different information and they have different behavior, and the simulation will be representing, in this case, how the cells from the immune system are attacking, in principle, the cells that are infected. In this case, the system is out of control, and the immune system is attacking not only the cells that are infected by the virus, but all the other cells too, and they are killing this model by exaggeration in the response, in the immune response. That is something we know that totally happen in real life. So this is still work in progress, but it's a way in which we can combine this molecular information, different cell types in in simulations for a given medical condition. That's the purpose. At the specific level, what we say that we are doing here, we are collecting information and processing information at the DNA, at the genomic level, this single cell information, for example, normalizing them and understanding that and creating with these profiles of individuals. The information about this specific cell from this tumor is specific of this tumor of this particular patient. At the same time, we are building the knowledge of how the human cell is working, how different components are interacting between them, and then we put these two things together. The generic knowledge about the functioning of the cells inform with the specific knowledge about individuals. Then this allows us to have different models, and then we can interfere and deduce and explore by simulation the different models in different conditions. For example, what are the different ways in which I can interrupt a given disease with different drugs that will be interacting with different parts of the system. So the two entries are models of how the system works and data, genomic data that can be used at the individual level or can inform us about what is going on in individuals. The challenge is to do this systematically, to do this accessible to the users, to the biologists, and to make it possible to do these simulations at a large scale. Because we are talking about simulating one of these organoids, that will mean a few millions of cells, each one of them with its own processes and its own simulations of the intracellular behavior. So we are, the processing of the omic data, the genomic data is by itself complex. The pipelines are, there's a lot of information, has to be processed, they are, I would say, requires a substantial computational time. The data is large, but on top of that, the data is highly confidential. So all these processes has to be run under the appropriate conditions of confidentiality, and this complicate all the operational work with the genomic data a lot. How do we compute? We try to compute close to where the data is, not to move these large quantities of data, and then all the cloud type of issues are related with this type of processing of the genomic data that we need as input of the cell-based simulation. The cell-based simulations, obviously, they are. We have a large number of cells. Each one of them with its own simulations. The parameterization of these systems is complex because they have many equations in the system, many different proteins interacting in the cellular system, many different cells interacting one with the other with different characteristics, and all this has to be parameterized. We are dealing with these cells interacting with them, but they are interacting in a given environment, that is the blood environment, that is a fluid, so there are issues of the interactions of the cells with the environment, where they are taking the oxygen and the nutrients. So the simulations has different levels, and they are complex, and we will see this in some more detail later. So that means that all the simulation requires high-performance computing. They are pretty demanding. They need systems that are connected. And we are also exploring the use of artificial intelligence for searching the parameters, for directing the simulations, and for analyzing the results, because otherwise these massive type of simulations are very difficult to handle. So that means that we see this as part of the toolkit of the Barcelona Supercomputing Center, all these type of simulations. A good part of the effort is machine learning to try to help in the search of the parameters and the simulations. And also the good part of something we will not be talking today is in the processing of natural language text, because part of the information that we are using is about the conditions of the patients, and this is coming from the electronic health records. That means that we need access to data. I'm not going to talk about data today because it's not the day for that. But all this area of biology, cellular systems, molecular biology, is very much linked to large-scale genomic projects, projects for understanding the genomes, understanding the diseases, understanding the marks on the genome, understanding the human brain, etc., means large consortiums generating large quantities of data that is confidential, has to be analyzed, and is the input that we use for the analysis or for the simulation. So a good part of what we do has to do with this large-scale consortium and the handling of data. Obviously, the other part has to do with computational resources. I'm sure that you have heard more about this these days. For us in Mare Nostrum, this is the current construction of Mare Nostrum. It's a powerful machine, one of the largest machines in Europe, and we are in the process of starting an even larger one, Mare Nostrum 5, that should be in operations next year, and it's really a jump in the scale. It's 10 times more powerful than the previous ones, and is entering into the exascale range of machines. Good part of what we are going to be discussing in the context of the PerMedCoE is how to adapt the different software to the different machines, because Mare Nostrum 5 will not be a single machine, will be a cluster of supercomputers, and that again brings another level of complexity, how these supercomputers are going to interact between them and how we are going to perform these simulations, adapting them to where is the data and what is the more proper architecture to perform these type of simulations. So that's, to skip that. So with this, we enter into PerMedCoE. So that was the scientific background, the opportunity because of the single cell technology, because of the organoid technology, because the supercomputer capacity, because of the large scale project producing a lot of data, for the first time in history, the opportunity to really make all these simulations at the cellular level something very significant for science and for biology. So that's the environment. This is how we started the HPC Center for Personalized Medicine. It's a part of the other Center of Excellence, one more of the European Center of Excellence, started by the European Commission. We are just at the beginning. We started 3 months ago. It's a consortium with many different centers. I have to highlight the participation of the other large supercomputing center that is in Finland, the CCSC, or friends and colleagues with a very interesting contribution to this dialogue between two supercomputing centers. Provide efficient and sustainable entry point for HPC exascale aggregate methodology. So the mission is to make accessible these methods in a way that the biologists can perform experiments on genomics and without much of a difficulty provide a simulation, execute a simulation of that will help him or her to interpret his genomic data. That's the goal. Obviously, for that, we have to optimize the software to be able to run in this platform. We have a number of use cases to do that, and we have to do this in the context of the developments of the other PerMedCoE, and there is a part of training, and then we want this Center of Excellence to be sustainable in the future. In practice, and Arnaud will go into the details, what we have done is, for starting, choosing 3 different types of software for intracellular simulations. One of them more dedicated to metabolic simulations, one of them more dedicated to control of intracellular processes, signal transduction pathways, and the other one more dedicated to stochastic simulations using a Boolean model. And then a system, one of the particular systems, a software, PhysiCell, for simulating cell-cell interaction, systems of cells. We are obviously collaborating with the developers of all these different systems, building the grounds for running them at the large scale in the supercomputing centers, and then at the same time putting this into workflows, and then discussing with the designers of the new processors and coprocessors what are the specific needs of this field and these simulations for them to be taking them into account in the design of the new processors. The use cases, without going into a lot of details, obviously, and I now will say something more about some of them. The obvious one is COVID-19, and I already showed you some of the initial work done in this area. Cancer diagnosis, and I already mentioned the possibility of using organoids for simulating tumors, tumor evolution based on these organoids, drug synergy, that is particularly interesting because we can use the intracellular system to try to understand how the action of one drug can be potentiated by using a second drug, and this is very important to avoid resistance, for example, in cancer. And then we have another case on rare diseases, when the link between the genomic information and the single mutations in single genes is easier to establish than in the case of cancers. Obviously, as I say at the beginning, we are developing this cell level and type of simulations, and this is very much related with the developments at the atomic and molecular level of PyXL. You can imagine that in the future will be interesting to link the simulation of specific proteins and specific drugs that will be one of the components of these pathways, that will be one of the components of these cells. And even more, we can imagine that these cell environments can be part of one of these larger scale simulation organs. So there is some connection between the other two centers of excellence, CombBioMed and BioXL, if we imagine these multi-scale type of simulations as something to be done in the future. And obviously we are seeing resources and collaborating with other centers of excellence, in particular with POP, for the optimization of the software, and with Hidalgo, because we are using the same workflow manager, or at least one of the workflow managers that we are using is the same one that was developed by Atos, or being used by Atos in Hidalgo, that is one of the other centers of excellence. So these are the partners. I'm going to enter into that. It's a combination of centers working in biology, centers developing the software for these simulations, the CSC, the Center Supercomputing Center in Finland, and a number of other partners, important, the MBLEBI, that is the main provider of knowledge and databases in the area of molecular biology. And of course, you are most welcome to visit the Barcelona Supercomputing Center. It's worth visiting. There is a virtual visit that you can make, but also as soon as it's possible, we would love to have you as a visitor at the BSC. So I finish with that, and I leave you with Arnaud, that will go now into the details and give you examples of the things that we are doing in different types of simulations at this level. And I'm sure that he can answer any of these questions, but if you have questions, more general question for me, and you want to ask questions now, we can take some minutes if there are some questions. And of course, my email is already in the presentations. I'm happy to also answer questions later. But if you have questions now, I'm happy to answer them.
Many thanks for your time. If any of the people in the call have any questions, you can ask them. If not, we can move forward with that now.
Well, if there are not immediate questions, I leave you with Arnaud. Arnaud, thank you.
Yes, thank you, Alfonso, for this very nice introduction. So let me share. How's this? Okay. If you can let me share the screen.
You should be available to share your screen.
Now, yes.
Vale. Perfect.
That's it.
Thanks.
That's it. Okay, so you should, are you looking at a presentation?
Yes.
Okay, Good. So yes, as I was saying, Alfonso introduced pretty well this project. So now what I'm going to do is to go a bit more into the details on some of the tools that we have used in PerMedCoE in order to tackle two different problems that are not small, like cancer and COVID. So I will go. I mean, you will see some of the slides will be repeated from the ones from Alfonso whenever I'm talking about PerMedCoE. But first, I wanted to do like a slight introduction on why we do what we do. So in fact, the many of you or all of you will know that at the beginning of 21st century we had this genome project, and we started to gather in biology. We started to gather huge troves of data. Huge amounts of data, either be from DNA or from RNA, also from other omics such as metabolomics, glycomics, proteomics, etc. And to all those multi-omics data, we had the trouble because before in biology we were used to study like one gene or one pathway, and it was very difficult to like come together with many of those data and to starting to analyze these data. So that was one of the reasons why, in order to browse or to interpret what this data meant, we started to work with networks and models. So of course, before this, networks and models were used in biology. So you have many models from the 60s, etc. But the boom in usage of these models started a bit with these omics data, and it was nice because thanks to these networks and models, we could integrate these omics data in order to infer mechanisms. So at the end of the day, we were not interested in that to saying to some patients that he or she has this mutation, but that mutation means something for the disease of this person. So now is not only to see that beta-catenin is mutated, but to see what is the consequence of this beta-catenin mutation. So you can see that if this is mutated, then something will happen. You will have like a trickle-down effect, and something will happen further down the road, etc. So, in our sense, these different tools that we were talking before, like Cobra, Selenium, MABOS, and PhysiCell, will be tools in order for us to build models in order to either test some hypothesis or to organize data in such a way that tell us something about the mechanisms of the system. So you may, I mean, as Alfonso said, these 4 different tools have slightly different focuses. So for instance, Cobra is used for metabolism, Selenopt and MABOS are used for signaling networks or for, let's say, pathways inside the cell. And then PhysiCell is used as an agent-based modeling framework to study cell-cell interactions or populations of cells. So depending on what is the different focuses that you want to study your cells, you will be prone to use one tool or another, or even combinations of tools. So for instance, PhysiCell, that is these population tools, can inside have as an add-on MABOS, or in the future it will be able to work with Cobra and even with Selenopt. So again, depending on the focus, you will need one to run another. And in fact, we have this I would say this background knowledge in biology that usually data leads to structure. So depending on what data you're working on, you will organize it in some given structure. So for instance, PPI network, which is protein-protein interactions, you will organize them with interaction network. With the only information that is there is that A interacts with B and with C. And for instance, activity flow is used whenever you want to use signaling pathways or you want to study gene regulatory networks. So in this case, A activates B or inactivates C. So you have these activation edges, and you can have a bit more description in these networks if you're working with process description. So for instance, this is a metabolic network in which A is converted to B whenever there's an enzyme, and this enzyme is modulated by another metabolite. And then at the end, you will have also entity relationship, which is like the finest granular level of description, in which you're not only saying that this modulates the enzyme, but you're saying of what is C affecting to. So if it's phosphorylating something, or if it's affecting another part of the enzyme. So again, data leads to structure, and then also structure leads to modeling. So you will not model the same way a PPI network than you would model a signaling pathway, that you would model a metabolic network. So for different data, you will have different structure, and also you will have different model. So so this is very important because the way you gather data restricts you to the way that you want to model this data. So the different biological knowledge will lead you to different types of modeling. And what is good in this perspective is that these the 4 different tools that we would use in our project are basically from two different kinds of modeling, which is nice because it allows us to tackle problems from different sides. So I will briefly talk about metabolic modeling, and I will talk about a bit about 3 of those 4 tools, so that you have a glimpse of how they can be used. So for instance, Cobra is used for metabolic modeling, and in our case with Omics data, we some of the Omics data can be used in order to build metabolic networks and metabolic modeling. So the first thing that we need to do is to build a network of the reactions. So reactions are conversions between a given metabolite and another. So you can concatenate reactions in order to build what is called a metabolic network. And this metabolic network, which is the association of the different metabolites that you have here in red, then you will with some constraints, you will be able to model, to have simulations on this. So the mostly widely used, or the widely used algorithm in order to simulate metabolic networks is called Flux Balance Analysis. That provides information on the reaction fluxes. So at the end of the day, you want to know if there is more flux reaction than in any other. And this is very interesting because sometimes you will be interested into overproducing a given let's say a secondary metabolite. Imagine that you want to have a beer that sends I don't know, vanilla. So you will be interested in rerouting flux towards the vanilla scent and less to the alcohol, because you don't want the beer to be I don't know, 20% degrees of alcohol. So this you can do analyzing reaction flux. You can study what reaction you need to constrain or what reaction you need to increase in order to have this rerouting of fluxes. And you do this by considering steady state. So the idea is that for the whole span of the life of the cell, the fluxes of reactions will not be the time span of the reaction will be very, very small. So you can consider that you have steady state of reactions, so the reactions don't change in time, but what will change is the proportions of the different reactions. So this you can look at it a bit more schematically in this way, in which this flux balance analysis first has this assumption of steady state and constraints. So you limit the possible solution space to this constrained biologically feasible space, and then you need to optimize a solution. Because usually, you talk with the biologist. If you give them a space of solutions, they will not like it. I mean, they would prefer to have like one point of that is the solution, not a hyperspace of solution. So in this case, this optimal solution is what we call biomass. So we optimize for the growth of the cell, and this biomass is just a mathematical construct in which we pick different metabolites of the cell that are representative of the biomass of the cell, and we optimize its growth. So if you see here, you have only the metabolic network, and at the end of the day, what you have is let's say the thickness of the edges would be the flux that is being carried in this reaction. So knowing what is let's say the wild type or the normal flux landscape, you can restrict different reactions in order to reroute these fluxes, or at least to browse what is the possibilities of these organisms. So this is what we call constraint-based modeling, because we use many constraints. So it's a bit different from kinetic modeling, which is, let's say, the ODE modeling that we can be more used to, in the sense that the kinetic modeling would have a complete description of the system. So you would need all the parameters of all the reaction, which is something that you don't have right now in terms of genome scale. So you have for some of the reactions more and more, but still you don't have for thousands of reactions. And then the solution is a unique. And in this case, we have incomplete information because we only have the stoichiometries of the reaction, and then the result is a solution space that, of course, using this optimality, you can restrict to one of the points. But constraint-based modeling has been widely used in biomedicine and also in biotechnology, and one of the things that is being used for is in order to personalize these models. So imagine that you have like let's say prior knowledge network, in which you know that this is all the possible connections of the different metabolites, and you use data from different tissues of a given human or from different patients in order to have liver-specific models or brain-specific models, etc. And then you can also personalize this model, not only with tissue but also with patients. So in this way, each one of those patients will have slightly different models, and each one of those different tissue will have also slightly different metabolisms, in which 80% of common reactions, but some of the reactions will be different. So this constraint-based modeling has been widely used in biomedicine, but also in biotechnology. So, for instance, all what is simulated with bioreactors or the use of, for instance, bacteria in order to evolve hydrogen is quite widely used in this field. So, if you want a bit more of information of this, CobraPy is the Python implementation of Cobra tool. So this Cobra stands for Constraint-Based Reconstruction of Analysis tool. So you have different flavors of it. You have the MATLAB flavor, which it was like the original one, and they derived in Python, and also they are starting now to work with Julia. And also, if you want to let's say study a bit more how this constraint-based modeling is used, you can just click here in this GitHub link, and this is a practical course that Miguel Ponce de Leon gave some years ago at the university, and I kind of like adapted it from it in order it to be a bit more intuitive or a bit more interactive. So you can just click, and a couple of clicks you have a Jupyter notebook, and you can just study this Cobra tool with the Jupyter notebook. So the second modeling that I wanted to talk today was about logical modeling. So logical modeling, as you can guess, is very useful when you have very scarce information, or that the questions that you're asking to yourself are qualitative in the system. So for instance, for instance, mutations or copy number, or presence or absence of different proteins, etc. So here the caveat is that you don't have information over time. So you don't have either on the data or on the simulations. So, and you don't have details about the precise biochemical reactions. So usually in logical modeling or Boolean modeling, what you have is that each variable can take two states, 0, that for us is readouts of activating and inactivating proteins, and you have Boolean logic. So you have logical rules that dictates the activation or inhibition of the given variables. So for instance, in this case, A is activated by C, and you see this green edge here, and then is inhibited by B. So this you write in this way. So the activation of A depends on the inactivation of B and the activation of C. And this you can see in this toy model is quite nice and quite small, but when things scales, it can be quite messy. So the way we simulate logical modeling is to find its attractors. So the attractors are a subgraph of the state transition graph. So let's say where the model evolves. So in this case, you can have either stable states. So for instance, in this case, is 0, 0, 0, the stable states. So if you look at the activation, if something activates B and B inactivates this, at the end of the day, you will have these stable states of 0, 0, 0. But whenever C is active, what you will have is a cyclic attractor. So you will have that the model will cycle among different states. So sometimes, or usually in biology, we prefer to work with stable states in the sense that we consider these phenotypes of the system. So cell growth or cell death or cell apoptosis or different ways of dying of the cell or of not growing, etc. But sometimes we also work with cyclic attractors, and typically the use case is the cell cycle. So the cell cycle of a eukaryotic cell doesn't stop until there's something that blocks the cell cycle. So this is typically a cyclic attractor. So this is two examples of models we have been working. So as you see, it can be rather small or rather, let's say, sparse, in the sense that here we have some 25 nodes and some 30 connections, but it can be quite dense. So for instance, we have here some 40 nodes and some, I think it was 100 interactions. So, and as you can see here, this is a convenient way to model signaling networks, or let's say regulation networks, in which you have different pathways that are representative of a given cancer, or in this case, it was, I think it was colon metastasis. And you can qualitatively see what happens if one node is activated or is inhibited, and what are the, let's say, the landscape of possibilities that the model has. So the typical questions that we ask whenever we're modeling Boolean logical modelings is, what are the solutions of the models, or what are the nodes that are more important than others of the network? How robust or sensitive is the model, and if we can predict genetic interactions, for instance. So what happens if I remove one node, or I force one node to be zero in accordance to another node, or what happens if both nodes are reduced to zero, if this is affecting much more the phenotype of the cell. So this is typically the truth table of our model. In fact, this is the truth table of this model here, in which on the rows what you have is the different variables, so the different nodes, and in the columns you have the different phenotypes, so the different stable states. And then we can analyze, okay, I mean, what happens if this ATP is forced to 1? So then I cannot reach these other phenotypes, but I can reach these others. So depending on the values of the different variables, you will reach given phenotypes or others. So the way we work with the logical modeling in our project is throughout this MABOS tool. And this MABOS is a way to have something intermediate between logical models and dynamic models. So what we use is to analyze stable state probabilistically. So for this we use a continuous time Markov process in the Boolean transition state space. So this is very easy to understand. Imagine that you have this toy model ABC, and then you have some kind of interactions between them. You can build what is the model states. So a model would be ABC, and these can have each one can have 0 or 1 value, and you have a way of transitioning from one state to another. So for each transition, you will have a parameter of a rate in order to go from 0 to 1 or from 1 to 0. And this parameter up and parameter down will be the ones affecting the probability of transitioning this Boolean state. So you have probabilities, you have some fixed time of evaluation, and you have some stochasticity. So you have the probability of reaching these different states. You have a Gillespie algorithm, or you work this with Gillespie algorithms, or with continuous time Markov process, which is basically the same in this case. So now the good thing is that now you can study perturbations in a probabilistic manner. So it's not what happens if I reduce, if I completely obliterate this node from 0 to 1, but what happens if I reduce the probability of this node to activate from 0 to 1 or to 50%. So now you can always relating to a wild type. Now you can work in a probabilistic manner how this model evolves. So in this case, this allows you to have stochastic simulation in which you may have different inputs to some given activation levels, and then you have stochastic trajectories of these simulations. And then what you retrieve is probabilities of reaching the different stable states. So you don't have zeros or ones, but you have 67% of reaching this state, or 10% of reaching another one. And now these you can do for all the mutants, and you can basically browse the different what happens in if you different input nodes. So imagine that here all the inputs are random initial conditions, so they can either be 0 or 1. So you have, let's say for this TNF input node, you have 50% of the trajectories start with 0 and 50% start with 1. And then you can compare these probabilities to what happens if you force one of those nodes to be 1. So you basically are restricting let's say the probabilities of the initial for this fat to only be 1. So in the case you can see here, so in red you have apoptosis, and in green you have survival. So this apoptosis that was like smaller than proliferating cells now is much more higher. So you have basically forced the cells, or encourage the cells to go to cell death. So a way that we have in order to refine these models is to personalize them. So as Alfonso was explaining, what we do is that we build these patient profiles with a given omics data, or we have single cell data, and then we introduce these, or we kind of like restrict the model values to depending on this data. So for this, you need to have like a mother model, or like a previous knowledge model. So in this way, we have like 3 different variables that we can touch in order to personalize this, and this allows us to have patient-specific models, or let's say group-specific models, etc. So for instance, we can compare the results of the model with the input data that we were using in order to constrain these patient profiles. And you can see that, okay, the input data has much more difference on the different subgroups of patients, but we still have the same order of patients in the model outputs in comparison with the patient profiles. And in fact, we can use the results of the models in order to have survival curves. And we see that we can differentiate the highly proliferating and the lowly proliferating patients, and also the highly apoptotic with the low apoptotic patients, using just a threshold to divide the patients into groups. So this way, this indicates that you can, I mean, the modeling, so the simulation outputs of the model has some value in the sense that it allows you to cluster the different patients into different subgroups of interest, such as highly in survival curve or in subgroups. So this we can do for patients, but we can also do with cell lines. So we can let's say constrain models, or we can personalize the models to the different cell lines. And now we don't talk about a prostate model, but we can talk about a prostate model of this specific prostate cell line. In this case, as you can see here, this is, for instance, the invasion output, and we have some variability of behaviors depending on the different cell lines. So again, if you want more information, you can read the paper, so you can just browse the code, and there are some examples and there are some templates that you can look, either for let's say how we analyze a logical model, or also how we can personalize these different models. So also, I'm not going into explaining what Selenapt is, but in any case, I let you hear some links in order for you to browse how Selenapt can help you, in the sense of if you have a good data that you want to use to constrain a model, because you're not sure if the connection between two nodes is and or it should be something else, you can use Selenapt for this. So here you have the two links from the code and from the description of the tool, where there are some tutorials and some how-tos in order to look onto how Selenapt is used. So my last part of the talk, I wanted to talk about agent-based multi-scale modeling. So you may know that agent-based is widely used in many areas of science, because it usually works with numerous agents. There is some heuristics on how we make decisions, and there is also the specific description of the environment and on the topology of the system. So you can find examples of agent-based in ecology, environmental science, and also in tissue biology. So in our case, you find many different kinds of agents. And in our case, we work with, in PhysiCell, we work with what we call overlapping spheres, or center-based. The thing is that here the cells are defined by a center and a sphere around the center. So in our case, the cells would be let's say all spheres, and we can work with the different conformations of this sphere. So we have in PerMedCoE, we work with PhysiCell, which is one of the base simulation frameworks. There are others, but this one is the one that we have experience and that suits our needs. But of course, I mean, you may find other examples that other frameworks may be useful, or other frameworks also in terms of Voronoi tessellation or vertex models, etc. So for instance, I know that there's groups studying the wings of Drosophila, of a fly, and for this they use vertex models, because for them center-based is not very interesting. So in this case, we have used PhysiCell in order to simulate spheroids of cells, which is this kind of setups in which what you have is spheres of spheres. So you have this is experimentally, you have some experiments in which you can have up to 2,000, 3,000 cells that make up this strawberry, if you want, or this blackberry. So the idea is that each one of those agents, or each one of those cells, have some kind of properties. So you have like different cell volumes, and you can have different positions, and you can also have different internal states, and you can also have different phenotypes for each one of those agents. And also you can have, as I say, the environment is very detailed. So you can have different voxelization of the environment, in which you can have 2D monolayers, so in order to have like petri dishes, or you can have 3D shapes in order to have like spheroid or ductal shapes, etc. You can even have a bit more complex shapes. So the idea is that PhysiCell first it has some mechanical equations and diffusion equations in order to solve what the environment, how the environment changes. So you can have like generation of oxygen on one side and it diffuses to the other side, or you can have generation of oxygen in all the the walls of the simulation. And these allow an environment that can be dynamic and also reactive. So you can, the cells can respond to the different levels of oxygen, or can produce something that then is transported into the environment, etc. So this allows you to have quite complex simulations, and also we call it scale because it has like time scales in which different things are happening. So for instance, you have the time schema of diffusion of the entities in the different environments, but you can also have the mechanical time steps in which the cell is moving or is being dragged or is being pushed by others. But you can also have the time step of the different phenotypes of the things happening inside the cell. So the cell dividing or the cell committing to cell death, etc. So what we did several years with collaborators was to use this multi-scale model in order to try to simulate the different phenotypes that we could see in cancer. So for this, what we did is merge PhysiCell with PhysiBoss, which is this stochastic simulator, in order to build PhysiBoss, which was a very, we find it very clever at the time, way of mixing two different names such as PhysiCell and PhysiBoss. But the idea now is that for each agent, what you have inside each agent, you have a Boolean model, and you simulate stochastically this Boolean model in order to capture different phenotypes that come from the different genetic state of these different agents. So in each one of those little agents, what you have is a Boolean network such as this, and depending on the inputs from the environment and also the genetic setup of these agents, different agents will have different let's say biological phenotypes. So in order to do this, we aggregated another timescale, which is the timescale of signaling, which is intermediate between what is happening in the environment and what is the commitment of the cell. So now what this allowed us to have different genetic and environmental heterogeneities. So before we could have already different tumor architectures with let's say bare PhysiCell, but with this PhysiBoss we can also now incorporate different cell strains, and we can all incorporate mutants, and we can incorporate different double mutants and look at epistasis of genes or synergies of genes, etc. So one of the experiments that we did with the PhysiBoss was to study drugs frequencies. So instead of having like a continuous drug present in the system, what you have is that each 600 minutes, for 10 minutes you add a drug, and then you remove it, and then you add and you remove it. No, you just wait the cells to incorporate them into their system. So in this setup, we found out that there was a given frequency where it was the only setup where the alive cells this drug was killing the cells, because this model has some particularities in which you can see that for more that you have a drug, the cells are resistant to the drug, and then at the end of the day you have even more cells than your starting point. And only by administrating these frequencies of drug, we could take this green line and just send it to zero. So in fact, we could look at how the cancer growth is diminished by changing the administration of drug, and this was done thanks to this PhysiBoss. So this is okay. Sorry. So this is the simulation, again, the simulation that we can build, in which you can have this was a video, but it's not showing anyway. So as you can guess, we can visualize the results of these simulations in many ways, and one of them is to have these figures, or also to have these videos. But yeah, so this allows us to use PhysiBoss for many different examples. One thing that I have not talked today is about migration of cells, but this is also something that we've been working on. So one of the things that we're currently doing with PhysiCell is to have migrating PhysiCell and PhysiBoss is to migrate into message passing interface, in which currently we know that if you work with, so this is in Mare Nostrum, and this is number of threads in another Mare Nostrum, and this is the parallel speed up. So if everything is perfect, the points should follow this line. And as you can see, the points are nowhere near to following this straight line. So this means that when you use 2 threads, the time of executing a simulation is halved. And then if you use 3, it's 3 times less than the 1. And if you use 4 threads, should be 4 times less than when we use 1. But this is not what happens. And in fact, what happens is that when you work with more than 6 threads, you see that the points deviate a lot from what it should be, or what the perfect speed up should be. And in order to solve this, and in order to have like higher, bigger simulations, what we have done is, together with a research engineer at BSc, Gorov, and also David Vicente, to incorporate message passing interface into PhysiCell. So now currently what we can do is that we can use the 48 cores of each node, but what we can do is to incorporate this OpenMP together with MPI in order to have a parallelized or distributed simulations in which different parts of the node, the different nodes will work together in order to have one huge simulation. So this will expand the scope of the simulation that we already have, and also will enable for simulations of complex behavior that right now we cannot do because we have a memory problems. So the way Gorov work is to divide the domain of simulations into subdomains, and that these subdomains communicate among themselves in order to let them know that there is a cell that has this domain or not, or that the oxygen is diffusing this way or this other. So each one of these subdomains will be handled by a different node, and this is something that is in the works. So we do have some results, but we're still working with Gorov and David in order to make this shine and with the newest features of the newest PhysiCell code. So this is one thing that we're working on, and another thing is to have different kinds of cells in the environment, so that each one of those agents to have like to be a different kinds of cells. So not only cancer cell, but also immune cells, or let's say non-cancerous epithelial cells, whatever. A third thing that we're doing is to incorporate metabolic modeling. So as you have seen with Cobra inside PhysiCell. So each one of those agents to have not only a logical modeling, but also metabolic modeling. And this metabolic modeling would direct, let's say, how the growth of the sphere is done. So again, if you want more information, I let you hear the different links of PhysiCell code that is developed by the main developers of PhysiCell are collaborators for Indiana University, and so let's say we branch out of it, and we're actively collaborating with PhysiCell in order to bring more features into this code. So lastly, if I have a couple more minutes, I wanted to talk about how the COVID use case that we have done. So the idea is that there is an ongoing development of PhysiCell for COVID, in which there are different virus infection and different aspects of the virus infection being modeled. And what we have done is to incorporate PhysiBoss in order to integrate different Boolean models. So we have started by an apoptosis model, which is cell death model, but the idea is to take advantage of any of the maps that have been developed in this COVID-19 disease map initiative. So so in March, April, and May, over 100 researchers will gather together in order to have molecular interaction maps built of what happens when the virus enters the human epithelial cell. So the idea is to take some of those maps to convert it into Boolean models, such as we have done here with apoptosis, using a tool by some colleagues of ours that are also part of this consortium, and to integrate these Boolean models inside PhysiBoss. So the idea is that, for instance, so for now we only have cell death, but the idea would be to have like different Boolean models of the immune cells, of the epithelial cells, even a virus Boolean model, etc. So right now we have already looked into this apoptosis model, and we have found like 2 different mutants, one of the epithelial cells and the other of the virus, and how these different mutants affect the spread of the COVID disease inside humans. So, for instance, another thing that we can work is with cells heterogeneity. So in this case, 50% of the viruses were mutated, and in this case, 95% of the cells were mutated. And you can see I mean, as you can see visually, you have different pictures. That means that you have different behaviors of the simulations. Or you can also see this in the snapshots and also in the temporal evolution. So yeah, so all of this was done together with colleagues of this project. So namely Institut Curie, inside the BSc also several groups, and also people at Heidelberg University and also UNILU. And also we have I mean, this is the rest of the partners of the PerMed consortium that also I mean, we haven't started to work with them, but we will at some point of the project. So if you have any questions, or if you need any more details on this, I mean, you can look at the PerMedCoE webpage, or you can follow us in Twitter, Instagram, or LinkedIn, and YouTube. And you can also send me an email, or you can also ping me in Twitter with my Twitter handle. So yeah, so I mean, if you have any questions, also, I mean, you can just ask it here. So thank you very much for your attention, and yeah, if you have any doubt, please proceed.
Many thanks, Arnau and Alfonso, for your time. We are glad to have you here for the first time in this course, and we are hoping to keep you having on the next iterations. It's a course that is generally held inside here at the BSc. So well, the next time is probably going to be more interactive.
Sure.
So if anyone has any questions, please feel free to make them now.
I see questions on the chat. So
yeah, I was just going to mention that.
Yes.
Sorry.
Yes, so Hamid asked about OneNote. So currently, in general, Mabos, Cobra, and PhysiCell, we use them only with OneNote. One of the first objectives of PerMedCoE is to scale these tools up to HPC and to exascale. So the developers of Cobra at Luxembourg University, they have started working with Cobra Julia in order to be able to use several nodes for their simulations. Developers of Mabos at Institut Curie have they have started now with using orchestrators, so using different ways in which to distribute the different Mabos simulations, and they will do let's say the MPI implementation in Mabos in a later date. And in our case, in PhysiCell, together with Gorov, we have already a beta version that can use many nodes, and we're now debugging this version because it has some troubles whenever cells migrate from one domain to another. For instance, last week we were working with integrating PhysiBoss inside Gorov code, and we have some troubles transporting the Boolean state. So this is something that we're working right now, and that will come soon enough. I mean, I cannot wait as well to work with many nodes in this kind of simulations.
Anyone has any other question? Well, if that's not the case, we thank you again, Arnau and Alfonso, for your time, and the rest of the people at PerMedCoE that were really helpful during the conversations we had a few months ago. So I hope you all the students had a good time during the course. And if I'm not wrong, I think that you will receive